it's a separate room by itself. Okay, so this is our cell culture hood. When you are not using it, the UV light needs to be on, which is on right now. This needs to be down, which is down right now, and the blower needs to be off. Okay, so everything inside is always kept sterile. Everything going in has to be sterile. Your cells, plates, anything have to be sprayed down with 70% ethanol. Only then do they go in. All tips, tubes, we have special pipettes for the cell culture room. They're individually wrapped, they're sterile. Use only these for cell culture. The way you switch the hood on is you first switch off the UV light, you switch on the blower, and then put the glass door up. Okay? Do not put the glass door up when you have your blower off. So what the blower does is it flows in air this way at very high pressure. So there's no chance of air from outside going inside. That's how it keeps it sterile inside. Okay? Everything that you need for cell culture is on these shelves. Please do not use it for any other purposes. We do not like to open up those tip boxes outside because they've been autoclaved. They are not like your ordinary tip boxes that are in the cupboard in the lab. These are autoclaved tips. Autoclaved PBS is right there. Autoclaved tubes are kept in beakers. I usually take care of autoclaving all of that stuff. If you're running low on anything, please let me know. I would definitely be happy to autoclave it. But like last time I wanted to use PBS and we were all out of PBS. So please don't let that happen because it takes a while to autoclave it, let it cool down, and then for me to use it on cells. So just let me know if you're running low on any of these things. Another thing that I would like to point out is the microscope. The on and off switch for the microscope is right here. A lot of times people look at their cells under the microscope, forget it, and leave it on. So please don't do that. These bugs have a very large half-life, but you still want to preserve them and keep them working. This is our 37 degrees water bath. If you want to warm up your media, you can do it here. We have 2 incubators. The top incubator is for all kinds of cells. You can use it for all kinds of cells. It's set at 37 degrees with 5% of carbon dioxide. The bottom incubator, if you're going to do any transfections with viruses, I would suggest use the bottom incubator because sometimes, if you're using viruses and you have it in with other cells, they could be cross-contamination, and you don't want other cells, your cells to be contaminated with viruses. Yes, that's it. Oh, trash. So anything that comes in contact with cells or any living material needs to go in the red bag waste, which is the biohazard waste. Any glass stuff goes into this. So if you use syringes, needles, sharps, go in here. And the normal trash, like wrapping paper for this, goes into normal trash. So make sure you segregate the trash. Do not put any biohazard waste in the normal trash. As a rule of thumb, anything that comes in contact with cells goes into the biohazard waste, so that there's no confusion. Any questions?
So can I use this dish directly for cell culture? It is direct?
Yes, it's sterile.
Sterile.
Yeah. But if you feel, sometimes what people do is they just leave it open. Today there are no open bags, but don't use them if they're open and the plates are loose.
Yeah. Use the sealed one.
Yes.
Not the open one.
One more thing, all the plasticware here is specially for tissue culture. So it should say tissue culture treated. Please don't use it for general purpose because this is more expensive than what the plasticware that we use for our normal stuff. So everything in this room is for tissue culture, just knowing sense. Just keep that in mind.
And a PCR machine shouldn't be left like this. If you're done with the day, you should turn off all the machine. If you're done with the reaction, turn off the machine. And especially on the weekend, we've seen a lot of times on the weekend people just left with this machine off. Because if you don't turn it off, especially at the end of the run, it's default set as 4 degrees. So the machine will be working hard to keep that temperature the whole weekend, and that significantly shortens the life of the machine. And when we replace it, it's a few thousand dollars. Okay.